Hello, hi there. We welcome you to the genetics series. In this series, we'll be looking at animal breeding and genetics. We'll be looking at various topics and how we can apply the principles underlying them in the improvement of animal breeding. This is the introductory module where we're going to introduce you to some basic things. And we want to start by looking at what genetics is all about. Genetics basically is the science of heredity, and its main concern is how characters are transmitted from one generation to another, or from parental generations to their offspring. And it has to do with the behavior of certain factors called genes. And genes are transmitted onto the offspring during reproduction. So we do observe differences and also similarities in animals, making reference to their genotype and the phenotype. These differences are attributable to differences in the manifestation of the genes controlling them. So when we have basic knowledge of these genes and how they manifest themselves, we can apply that in a desire to improve the productivity of farm animals. So we have four major branches we are concerned with: Mendelian or qualitative genetics, population genetics, quantitative genetics, and also molecular genetics. So Mendelian genetics is fashioned out of a series of experiments carried out by the monk Grigor Mendel while working with the garden pea, Piscium sativa. He's worked with this pea because this pea is easy to work with, and they have certain contrasting characters that make it easy to follow a trait. And he considered simple traits that were more or less alternatives, like tall versus short, yellow versus green, axial flower position versus terminal flower position. And these traits are controlled by very few genes. So, and this has led to the branch of genetics called qualitative genetics, which deals basically with qualitative characters. The attributes of qualitative characters are that they are easily measurable, are not easily measurable. They are discontinuous in nature, that is, they do not vary very much. Green is green, yellow is green, is yellow, animals with horn, animals without horns. Uh, so this makes them to be discontinuous in nature. They are also discrete. Animals either have them or they do not have them. Animals, for example, either have horns or they are polled. And in this trait, animals can easily be classified into more or less distinct classes. We don't have much overlapping, and basically because they are controlled by very few genes. The environment has been found not to have much influence on the manifestation of qualitative traits. So the influence of environment is highly minimal. Here we have some examples of qualitative traits in farm animals, like the presence or absence of horns, male or female, skin color, plumage in chicken, polydactyls in man, and also in chicken. These are some few examples of qualitative traits that are found in farm animals. So Mendel basically worked on monohybrid and dihybrid traits. Monohybrid meaning trait controlled by one gene, and dihybrid traits controlled by two genes or more, mostly two genes. And these traits are under very control of very few genes, or a few at most. So population genetics describes in mathematical terms the consequences of Mendelian inheritance in the population. So it's more mathematical in nature, and here we are applying Mendelian principles to evaluate gene frequency changes in population, and through this we can calculate the gene and the genotype frequencies in population of animals. The fallout of population genetics is what is known as Hardy-Weinberg's law. And this law basically states that in a large population of random mating individuals, the gene and the genotype frequency is expected to remain constant from one generation to another in the absence of selection, mutation, migration, and genetic drift. So the basic keywords: large population of random mating individuals, gene and genotype frequencies remaining constant one generation to another when there is no selection, mutation, migration, and genetic drift or chance. Now, this law is very difficult to hold. However, if we can maintain the following conditions below, then Hardy-Weinberg's law or equilibrium is expected to hold. One, the population must be very large, because when you have a very small population, the chances of not holding this law, or this law not holding, is more than when the population is very large. But mating must be at random. When mating is at random, it means that all the animals will have an equal chance of mating or reproducing, and hence leaving behind their offspring. In such a population, there should be no selection. No member of the population is to be unduly favored, because when you select either for feeding or for reproductive purpose, you are giving favor to that particular member. Mutation is not expected to occur in this population, and where mutation occurs, both the forward and the backward rate must be equal. Likewise, no migration is expected into or out of this population, and if it should occur, the rate of immigration should equal the rate of emigration. The population is expected to be balanced at any particular time. And finally, the trait concerned must not be sex influenced. So in such a population, if it exists, and if you have a gene with presented by these alleles, then the frequency of the A allele is expected to be A frequency of A, and we can represent this by the letter small p, and the frequency of the recessive allele is given by this, and we can represent it by letter small q, such that when this population is at equilibrium, we expect that p plus q should give us 1. The implication is that p is half of the entire frequency of genes in that population. So p should be equal to q, and this is equal to 0.5. It also means that when we have p, we can find out what q is, and if we have q, we can find out what p is. So if these alleles in this population interact in this way, in the F1 generation, we expect to also get these genotypes. So in such a population, then the frequency of the homozygote dominant AA is equals to p squared. Why is it p squared? Because there's double homozygote dominance, so it's equals to p squared. While the frequency of the homozygote recessive small a should be q squared, and then the frequency of these two is 2pq. So the implication is that if this population is in genetic equilibrium, then the genotypic frequency will be p squared plus 2pq plus q squared, which should give us 1. Now, with this at the back of our minds, it's easy for us to calculate the gene frequencies, and this we can do using 2 different methods, either by the direct count method, and this is applicable where we are sure of the actual genotype of the animals that we are sampling, or by the square root method when we are not so sure of the actual genotype. And so we estimate this from the recessive gene only. So we want to take examples of how we can obtain gene frequencies either by the direct count method or by the square root method. So in our example, we have taken a count on a population of 1,000 cattle, and here we discovered that 200 of them were red in color and represented by this genotype. 500 had brown color represented by this genotype, while the remaining 300 were white in color represented by this genotype. If you add this, you realize that you have 1,000. So we want to determine the frequency of both the red and the white genes in this population. So using the direct count method, we'll bring out first of all the observed number. We have 200 of this, 500 of this, and 300 of this. These are the alleles, and we want to estimate the total number of alleles, capital W and small w, in this population. If we add this, we get 1,000. So in this genotype, from this number of animals, we have 400 number of capital W alleles. And why is that? It's because we multiplied by 2 because the gene is in the double zygote state. Small w is not counted because it's not existing here. Here we have 500 cattle with this genotype. And since we do not know which one exactly is carrying this or this, then we split it in between. So we have capital W 500 and small w is 500. Here we have 300, there's no capital W, we have 600, again multiplied by 2 because it is in double zygote recessive state. So the total number of genes in this population is 2,000, and this is obtained by adding this or by multiplying this by 2. This is a way of balancing out 2,000. So the number of W is 900, 400 plus 500. The number of small w is 1,100, which is 500 plus 600. So to obtain the frequency of capital W gene, allele rather, is 900 divided by 2,000 will give us 0.45. And the frequency of the small w is 1,001 divided by 200, which is given. If you add these two, you realize that we have P plus Q is equals to 1. But we want to see if this population is in Hardy-Weinberg's equilibrium. So therefore, recall that P squared plus 2PQ plus Q squared equals to 1. So our Q squared, P squared, is the frequency of the double homozygote allele, and that is 0.45 squared. We get this. For the recessive allele, it's also double homozygous, 0.55 squared. We get this. And 2PQ applied here give us 0.495. So now we can check if this is actually correct, and meaning it is in Hardy-Weinberg's equilibrium. And for us to do this, we use the tool known as the chi-square test. And this square test, we use it to estimate. First, we have to estimate the expected number of individuals from each genotype. What was counted, we want to be sure whether it is what is actually expected. So for the capital W, capital W alleles, that will be 0.45 squared times 1,000 will give us 202.5. For the small WW, 0.55 times 1,000, the squared rather times 1,000 will give us 302.5. For the heterozygote, you recall that is 2PQ with 0.495, so multiplied by 1,000 was 495. So the next stage is to bring our values into this table. So this is the originally observed number of individuals, totally 1000, and we have estimated it that this is actually what is supposed to be in the population. So we are going to subtract the observed from the expected, and that will give us these values. So to go ahead, we have to apply the chi-square statistics, and the formula says that the sum of the observed number of individuals minus the expected number of individuals squared divided by the expected number of individuals. This is a shortened form of the same formula. So we're going to put our values into this formula. We have minus 2.5 squared plus 5 squared plus minus 2.5 squared divided by the expected number of individuals, and if we solve this, we get 0.103. So the next thing we want to do is to compare this value with what is in the chi-square table at 2 minus 1 degree of freedom, and that value is 3.841. So in arriving at conclusion for testing whether this population is in Hardy-Weinberg's equilibrium, we have to take note of these two values. We'll have observed that the difference between the observed and the expected value is not significant. And how did we arrive at this? And this is because the chi-square calculated is actually less than the chi-square tabulated. If this value was higher than this, then there would have been a significant difference. So we can actually conclude that the null hypothesis, which stated that population was in Hardy-Weinberg's equilibrium, will accept it, while the alternate hypothesis, which was saying that the population is not in Hardy-Weinberg's equilibrium, will be rejected. So this is how we can use the direct count method to evaluate if the population is in Hardy-Weinberg's equilibrium or not. Now, the second method is known as the square root method, and this uses the formula q squared is equals to square root of m divided by t. Let's define these values, these parameters. So q here is the frequency of the recessive gene, for example, of such recessive gene as spore gene, albinism, and so on, while m is the total number of individuals having that particular gene. Say this is 386, we want to use these values for our example, and t is the total number of the entire, in this case, local chickens that were sampled, all genotypes, and let's say we are using 1039. So if 386 had spore gene, which is recessive, then the balance between 386 and 1039 are those that are sporeless. So applying this formula, we get that q is equals to square root of 386 divided by 1039. And if you do that, it will give you square root of 0.372, and that is equals to 0.16. So it means that the frequency of the recessive gene is 0.61. And if P represents the G frequency of the dominant gene, it means we can get it from the formula that says that P plus Q is equals to 1. Remember, we said that if we have Q, we can easily get P, and if we have P, we can easily get Q. So in this case, the frequency of P is equals to 1 minus Q. Apply that into the value, 1 minus 0.61 is 0.31. Therefore, the frequency of the dominant smallest gene is 0.39 in this particular population that was sampled. So again, we're going to use chi-square to try to test if there's a significant difference between the observed and the expected, which is when under normal condition. And again, we are testing two hypotheses. The null hypothesis, this states that there is no significant differences between the observed and the expected number of birds. And the alternate states that there is significant difference between the observed and the expected number of birds. So in our example, the observed number, 386 of them were spored. Those without spore were 659. So now, because the absence of spore is dominant in poultry, and we assume that this population is a random mating one, we will expect that about 75% or 3/4 of the total fowl sampled did not have spore. Meanwhile, those with spore, that's the recessive gene, we also expect that 25% of them will have that. And this is because we expect that this population is not just a random mating one, but because the traits are following Mendelian ratio of 3:1. Therefore, the expected fowls, it will be 3/4 multiplied by this will be 779.25 that are having spore, and 259.75 that 779.25 that do not have spore, and 259.75 that are having spore. Remember the formula for chi-square statistics: observed minus expected squared divided by the expected. So we bring that into the equation. We have this was the observed minus this was the expected squared divided by the expected. That's for those that are having spore, and these are the ones that are not having spore, and that will give us an answer of 81.817. This is the chi-square calculated. So if we continue, want to take a decision, recall that if the chi-square calculated is greater than the chi-square tabulated at 95% confidence level, that is at 0.05 level, we are expected to reject the null hypothesis and accept the alternate, and vice versa. And from our example, the calculated is 81.817, which is way greater than the tabulated at 95% confidence level. So therefore, because this is what is obtainable, we are going to reject rho, the null hypothesis, and accept the alternate hypothesis. So we conclude by saying that there is significant difference between the observed and the expected number of fowls. So in this particular population, the Hardy-Weinberg's equilibrium law is not being obeyed. So something is wrong, meaning that there might have been cases of selection, there might have been fake cases of immigration from a neighboring farm, or there could have even been cases of a mutation that occurred without anyone knowing, or possibly that spore is a sex-influenced trait. So quantitative genetics is a direct opposite of qualitative genetics, and it deals basically with traits that are measurable. We refer to them as metric traits, and most of these traits that are measurable usually fall into the economic traits of farm animals. So these traits show what is known as continuous variation, and what this simply means that different animals, we assume any value between the two extremes. The two extremes here we mean from the best to the worst. Many animals can assume any value within that. So we say that this trait show continuous variation. And Unlike the qualitative traits earlier treated, these traits are very easily influenced by environmental factors such as nutrition, climate, disease, and the management of the farmer. If these factors are not well managed, it will influence the economic traits or output of the farm animals on a farm. And also these traits cannot be easily classified into distinct classes, as was the case in qualitative traits. And again, these traits, unlike quantitative traits, are controlled by several pairs of genes, so we call them polygenes. And what happens is that each of the genes that control these traits contribute something to the final phenotypic value of a trait. So for example, we will say that milk production, for example, in cows is controlled by several genes. Could be feed intake gene, could be gene that controls water intake, the production of the milk itself, heat control, and so on. So all the genes that play a role in the production of milk play their little roles, and each of those roles are added up to give us the phenotypic value, which is the quantity of milk that we measure in the animals. So these are some other examples of quantitative traits. There are many. See the milk, wool, and egg production, body weight, feed intake, egg weight, egg number, and several others. So molecular genetics is the rave of the moment. It's also referred to as genetic engineering or biotechnology, molecular biology, as the case may be. And what it does is it applies recent concepts and recent discoveries and knowledge of the nature and the property of the DNA and the gene that is found on the chromosomes. And these recent discoveries and knowledge and concepts, when applied in the right way, have been found to lead to improvement in livestock productivity. So these are some of the methodologies used in molecular genetics, like cloning technology, recombinant DNA technology, gene therapy for repair of rogue genes and so on, vaccine production, marker-assisted selection, genomic selection, and so on and so forth. This is the rave of the moment, and several things are happening in this area that is helping us in the genetic improvement of farm animals. So we have come to the end of the introductory aspect of this module. Feel free to subscribe to this channel so that you can get notification, share with others, write a comment, and please like the channel. I will see you as we continue.